Hello, everybody, and thank you for registering today or listening today. As mentioned, my name is Ashleigh, and I'm Director of Business Development for Intertek Pharmaceutical Services in the UK. Prior to moving into business development, however, I have spent over 25 years working in the area of biologics characterization and quality control. In the next few minutes, I'm going to give you an introduction to some of the quality control analytics required to support inhaled and nasally delivered biotherapeutics programs, really tailoring this from the biologic side rather than from the formulation or device development. So, Intertek is a huge global organisation, a section of which is dedicated to the provision of analytical solutions to support the pharmaceutical industry. Our team has over 30 years' experience in both biologics analysis as well in oral and nasally development programs, and the skill sets we've brought together to support this exciting area of discussion today. So why is inhaled and nasal delivery such an exciting prospect for biologics? There are several key advantages to these delivery routes, including target delivery to the lung, as well as providing an efficacious dose route for systemic delivery, a point which can be a particular challenge from some advanced therapy types. Although today's presentation is mainly concentrating on solutions specific to protein-based biologics, such as hormones and monoclonal antibodies, many of the fundamentals that will be discussed are also equally applicable to other advanced modality types, such as peptides, vaccines, cell and gene therapy products, as well as DNA or mRNA-based medicines. And for example, the potential of the nasal or inhaled route for vaccine administration has never been more evident than in recent months, with the race to find a viable COVID-19 vaccination for mass administration, where both nasal and inhaled formulations are currently widely under development, as a snapshot of which are shown on the screen now. So comparing the advantages and disadvantages of nasal or inhaled delivery. Firstly, large molecules can be innately difficult to deliver systemically, as mentioned previously, at an efficacious dose level. An intranasal route avoids some of the enzymatic degradation and first pass hepatic elimination, which really reduces this barrier and it can generally are what limits the biologics absorption and therefore it can increase this property, contributing to fast onset of action. More so when considering some specific indications such as treatment of Parkinson and Alzheimer's. Several biologics saw promise in this area as therapeutics, however require delivery into the central nervous system for action. Nearly all biologics substances are severely restricted from crossing the blood-brain barrier under normal conditions, and therefore delivery of these species via the nose to brain route therefore presents a real viable solution, bypassing the problems of blood-brain barrier um, cross. Improved patient experience and the ease of administration are both additionally significant benefits. Given the tendency for the enzymatic degradation that we mentioned in the GI tract, the current typical route of administration of biologics is via subcutaneous injection or IV infusion. And these are not only less comfortable routes of administration for the patient, but it also require the therapies to often be administered in a healthcare environment. And this reliance is significantly reducing the size of the global population that can potentially gain access to this type of medicines, but also in the logistics of administration. For example, if you go back to the kind of the global COVID administration or COVID vaccine administration, it'd be much simpler to do if this could be delivered in a nasal spray. On the downside, working with a biologic in general presents many challenges, and not excluding those of the analytics we're going to talk about. And these are things such as quite often more material being required, and this material is often incredibly expensive. There's often a greater need for troubleshooting in the development and to actually gain stability of the material. And this might necessitate the need for atypical additives to be used, for which, in the worst case scenario, there's no tox profile by a nasal or inhaled route. And many of these factors combined can often mean that the additional development and drug requirements simply make this type of administration financially unattractive. Once the decision has been made to develop a nasal or inhaled biologics, there are effectively two product profiles or two areas that we need to really consider through the product development in and implementing the QC strategy and the methodologies in specific needed to control. First is consider the product as any other inhaled or nasally delivered material. And this not only means in choosing the most suitable device and designing the right formulation, but importantly in the establishment of a core set of analytics to actually enable monitoring of the product, a lot of which are shown on screen. And although these are not often simple to establish, the requirements are well understood and the choice of parameters to measure are defined in regulatory guidances such as USP 601 and 1601 relevant nebulizers, which really look to cover the release stability and product characterization. Where perhaps the new big challenge comes when considering biologics in this space is the actual introduction of analytical methods to assess control pertinent to the biologic itself. To understand what this additional scope means, we need to first think about what is a biologic. In this case, what a protein is for the specifics of this presentation. So proteins themselves are a range of different species such as hormones, monoclonal antibodies, globular proteins, and they really, although they can massively differ in size, they all kind of follow the same chemistry and the same properties. When we're considering these type of products, there are really two top-line messages moving from the small molecule world. Firstly, that CVs and acid performance of less than 5% relative RSDs and accuracies are less than 5% are no longer a thing. Biologics methods are inherently less accurate and precise, with values of 20–25% not being uncommon. Secondly, dosages in these type of forms are not necessarily just based on the mass of a protein or the mass of a drug delivered. It's actually based on the activity of a drug delivered, and this is something that can change batch to batch on a weight basis. So proteins themselves, when we consider the structure, are polypeptides comprised from amino acids. When you have greater than 40 residues in a chain, this is where you class as a protein. When it's less than 40, it's classed as a peptide. So the sequence of amino acids in the polypeptide chain is defined as the primary sequence of the protein. Hydrogen bonding that occurs between hydrogens and carbonyl oxygens in the chain leads to, amide hydrogens and carbonyl oxygens in the chain leads to the protein to form as what we class as a secondary structure, which is often alpha helical or beta sheeting form. This secondary structure then goes on to fold on itself to give what is known as the tertiary structure. So this is what gives the protein its overall 3D shape and is the most stable form of the protein. And this is defined as the native state. And it's really important to actually always remember that it's this 3D structure, this native state of the protein, which actually gives it its function. Finally, we need to think about a quaternary structure, where a number of these native tertiary subunits join together. And this can be a natural desired phenomenon. For example, hemoglobin is actually made up of 4 subunits, but also can be very, very much undesirable in the case of aggregate formation, which is a key attribute that needs to be monitored, as it can not only reduce the efficacy of the product, it can actually greatly reduce its safety in terms of a much, much larger risk of an immune response. When considering product development, therefore the major challenges is to develop a product and a manufacturing process which maintains a protein 3D structure, which inhibits any aggregation, and doing all this while not compromising the product potency and safety. And really this is not an easy task. Protein structural integrity is based on relatively weak interactions, which unfortunately makes the material susceptible to change in structure. So degradation and aggregation can be caused by many features such as changes in temperature, agitation of the material, interaction of the container or the system, to name but a few. And all of these have to be thought about when actually considering the design of the device, when considering the components of the formulation, as well as the actual testing regime. For example, if we apply all this knowledge we've just learned about the tendency of a protein to degrade and what may actually induce degradation or aggregation, and we overlay this with a typical lyophilized drug to nebulizer to PMDI or dry powder inhaler type production, we can see a number of areas where potential degradation or aggregation can occur. So if we move on to specifically consider those analytics that need to be in place above those that would typically be in place for a nasal or inhaled product, the majority of requirements fall into three areas. Methods are required to assess aggregation, degradation, and potency. So degradants typically would include in this sense assessment of charge variants such as oxidation products, and the methods to actually assess these species, although complex, are well documented and include things such as a capillary electrophoresis, an ion exchange chromatography, or Hillick chromatography. Aggregation and potency, however, are something which require a bit greater level of understanding and something I'll come on to talk a little bit more. The final thing I'd like to specifically talk about is the assay. So although an assay to measure, for example, delivered dose is something that will be common to all products, there are certain challenges for the quantification of a biologic that really warrants further discussion, and I'll come about to talk about that in the rest of the presentation. But one major observation is due that to the inherent challenges of analysing biologics in terms of the accuracy, precision, and robustness of the methodologies. It's really typical and even expected from regulatory perspective that orthogonal approaches are used to monitor the same critical quality attributes. So the techniques that I've detailed on this slide are those typical methods that are used usually in combination to assess the properties that we're talking about. The larger the bubble, if you like, the most widely applied in each area. If we move on to consider the first of these, an aggregation, it's a real expectation that this will be assessed using a stationary and a dynamic approach. For example, this may include a combination of both size exclusion chromatography and dynamic light scattering methodologies. There are, however, many disadvantages and advantages of each of the approaches that you can use, all of which I've kind of highlighted on screen for the main techniques. And as an example, SEC has significant advantage in its use and it produces a quantitative result of what is acceptable accuracy and precision, using techniques and equipment that is methodology that is common to most laboratories. However, the interaction of the product with the column, the solvent, it can actually lead to low recoveries of material or, importantly, the formation of aggregates giving false results. So during method development and validation, it's therefore really typical if SEC is to be used as the final release or quality control method. Other analysis has been performed by something like AUC, which is an in situ technique to actually check out the process of separation isn't inducing any aggregation or low recovery. As I mentioned, quantification of a protein as a delivered dose is something that on first observation may appear incredibly simple. For assays of proteins, it's not typical that chromatographic approaches akin to small molecules are used, but rather concentration is determined using a non-specific total protein assay in its simplest form, something as absorbance at 280 nanometers. Here you use the absorbance at 280 nanometers with a validated or calculated extinction coefficient to convert absorbance to concentration. So this approach is very much the go-to method for most protein quantification programs, unless there are specific issues concerning matrix interferences, specifically if you've got another protein in your formulation buffer, or in this case where you need a specific protein assay, you need to move on to something much more complex such as ELISA or LCMS, which although give you that specificity are really quite complex to develop and again can be quite prone to quite variable accuracy and precision. Assuming, however, that a suitable quantitative method is established, one of the greater challenges can actually be determining the delivered dose might be to do to inherent tendency of proteins to absorb onto surfaces, for example, the surfaces of the device, for the surfaces of the sampling equipment. The extent of adherence that you actually get is specific to an individual protein, it's specific to the protein concentration and the surface on which you're actually dealing with. And any slight changes to one or more of those can actually lead to differences in the extent of adhesion. There are several ways to avoid this. For example, you can use specialized products in process where possible, or you can pre-coat any surfaces with a surfactant or a blocking protein. So this is something like BSA, which will effectively flood the surface. Similarly, these materials could be actually added to the extraction buffer. However, depending on the approach that you have to use to recover the material and the actual components that you are including in that process may actually drive the assay method that you're going to have to use. For example, if you use BSA to actually coat or to extract protein in the delivered dose method, the A280 or the colorimetric approaches won't work, and you'd need to have an ELISA or an LCMS approach established. The final method that I really wanted to describe, and in terms of its implication use, is that to achieve potency. So, potency is in itself is something that cannot be directly correlated to the physiochemical attributes. And an extreme example, you could achieve 100% delivered dose, but a product of zero efficacy. How to measure potency is, however, well defined by industry and regulators, and as an example of 21 CFR 610.10 guidance, as described as being dependent on the in vitro and increasingly less typical in vivo tests. As a word of warning, going back to the kind of differences in acceptance criteria, potency assays are based on a cellular response. As such, they're really prone to inherent variability. And the very best assays work at percentage relative standard deviations of 20% or higher. Often you can go up to 25%, 30%, and still be the very best you can get. Potency assays are complex and probably the most complex across all different types of quality control methods, regardless of modality or purpose that you can actually put in place. What I've tried to do here is present the very simplest definition of what an actual potency assay is, how it works, what it does. But I really apologise to any cell biologists in the audience, as this is perhaps the most oversimplified thing in the whole entire world. So in the very most basis, a cell line is chosen based on an alignment or based on the mode of action of the actual product, which is cultured in a laboratory setting. The target test material is then applied over an 8 to 12 point dilution series, which actually is there to affect one of the more biochemical processes in the cells, which will either reduce or increase the expression and therefore the concentration of a target or marker protein. This target or marker protein can be quantified by ELISA, and from this and comparison to a reference material, the potency or the relative potency established. So calculation of potency is really based on comparison of the response of the test material to that of a well-defined reference material. So this reference material is something which has been well characterized and typically would have been used in a clinical setting. The response of the two are plotted, the parallelism is compared, and then the relative potency based on something like comparison of the EC50 of both materials. This is often a challenge, and as I said, acceptance criteria and acceptance limits are often very different to what would have been seen in typical chemical assays. However, understanding that you're keeping your potency through nebulization, through nasal formulation, is really probably one of the most important features that you could possibly address in these type of products. So in summary, there are real advantages to nasal or inhaled delivery for biologics, specifically in this instance proteins, and advantages that could overcome some of the challenges that are seen with achieving systemic bioavailability, or in getting the product to the target areas such as the central nervous system. Product development strategies therefore require establishment of not only those methodologies that are traditionally associated with nasal or inhaled products to support development and ensure quality through development, release, stability, etc. But also you need to include those methods that actually address the quality control attributes of the biologic itself, so specifically aggregation and potency, as well as some of the degradation methods, such as the ones we talked about for oxidation products. These methods are inherently less complex and less accurate and precise than their small molecule counterparts. And often there's a requirement to use orthogonal approaches to ensure the very best quality and consistency of product. These methods are often quite time consuming to put into place and really something that needs to be thought about very early in the development program. So thank you very much. Thank you for listening, and I'd be delighted to take any questions.